All right. Well, good afternoon, everybody, and welcome to the second Retina Australia webinar for 2021, an update from two of our Retina Australia grant recipients. My name is Sally Turnbull. I'm the Administrative Officer for Retina Australia, and I'll be the facilitator of this webinar today. There has been a late change to the speakers for this afternoon. Unfortunately, Andrea Vincent was unable to join us due to a family illness. However, Associate Professor Lauren Ayton has very generously agreed to step in and provide an update about some of her current projects. So now I would like to introduce the Chairman of Retina Australia, Mr. Leeton Boyd, who will say a few words of welcome.
Thank you, Sally, and welcome to everybody. It's very exciting, these webinars, and it's good that Sally's been able to arrange these. Thanks to Raymond today, and it is quite a shame that Andrea, who's been one of our long-time supporters in Retina Australia over a very long time, was unable to join us. But Lauren Ayton, who's been certainly a long-standing supporter, friend of, and most of you would know Lauren, and Lauren at very short notice has agreed to fill the void, and we're so grateful for Lauren for doing that. So without further ado, I'll hand back to Sally and enjoy the afternoon. Thank you.
Thanks very much, Leeton. Okay, so now I'm just going to introduce Dr. Raymond Wong, who's a Principal Investigator at the Centre for Eye Research Australia, the University of Melbourne, and a Guest Professor at Shenzhen Eye Hospital, China. He is a stem cell biologist with 18 years of research experience, specialising in cellular reprogramming and stem cells in eye research. Currently, Dr. Wong's research focuses on understanding the genetic signals that define retinal cells and using cell reprogramming and stem cell techniques to study and treat retinal degenerative diseases.
And thank you for the kind introduction. And I will take this opportunity to thank Retina Australia to organize this webinar so we can present our research here. So today I'm going to talk to you about our work in developing a gene therapy to regenerate photoreceptors. I'll start by introducing our team here. So my name is Raymond Wong. I'm a stem cell biologist by training, and I was joined by two very talented PhD students in my lab, Crystal and Daniel, who work on this project. And we're supported by many clinician scientists within the Centre for Eye Research Australia, including Keith Martin, our Managing Director, who brings in expertise in gene therapy, Alex Hewitt, who brings in expertise in CRISPR technology, Chi Liu, who brings in expertise in animal models for retinal degeneration, as well as Tom Edwards and Robin Geimer, who specialize in running clinical trials for gene therapy and retinal degeneration. So I'll start by saying that blindness is a global problem, and in particular, the impact of blindness associated with retinal degeneration. I think this audience is fairly familiar with this concept. This is what the retina looks like. It's really a thin layer of cells at the back of our eye that consists of different types of neurons. How our vision works is that the photoreceptor in particular that you can see here are the light sensing cells that pick up light signal and convert them into electrical signal that get transmitted through multiple neurons and ultimately to the optic nerve cells, and that get carries from the eye to our brain to interpret as an image. Now, given the importance of photoreceptors, it's very obvious that if there are photoreceptor degeneration and they start dying, it will obviously cause a very huge problem in our vision. Now, in fact, there are many blinding diseases that are associated with photoreceptor loss, including retinitis pigmentosa that affected more than 1 million people worldwide, age-related macular degeneration that affected 190 million people worldwide, Stargardt disease that affected more than 780,000 people worldwide, and actually many more, including cone rod dystrophy and so on. The key problem that we're facing is that there's really no cure for blindness once the photoreceptors are lost. And our research is trying to address that by developing a gene therapy to regenerate photoreceptors that could potentially treat multiple degenerative eye diseases. So how does gene therapy work? Well, in a inherited retinal disease, a mutated copy of a gene would start causing the retina cells to die and degenerate. Gene therapy works by taking a healthy copy of a gene and packaging them into a virus called adeno-associated virus, AAV. And this can be injected into the eyes, and that would effectively introduce a healthy copy of a gene to replace the faulty copy that causes retina degeneration. Now, in fact, currently there is a clinically approved gene therapy in the eye. This is a drug called Luxturna. This is the first clinically approved AAV gene therapy for retinitis pigmentosa, and it works by introducing a healthy copy of RPE65 to replace the faulty copy in the retina. Now, the problem with this is, although it is a very exciting development for gene therapy, this is only suitable for a small number of retinitis pigmentosa patients, about 1% or less of all the retinitis pigmentosa patients. And it's really not suitable in cases with extensive photoreceptor degeneration. So our solution is that we are developing a gene therapy to regenerate photoreceptors that could potentially help all retinitis pigmentosa patients. This is really our game plan of trying to develop this gene therapy. What we want to do is to develop a treatment that consists of an injection of a set of reprogramming genes into a vision impaired patient. And these genes would target the stem cells within the retina, and these are called the Mueller glial cells. And using a technology called cell reprogramming, we could then turn the stem cell and stimulate them to regenerate new photoreceptors, and that would effectively restore vision. So let me explain why we want to target the Mueller glial cells. Well, it turns out there are very interesting cell types within the retina. As I mentioned, they do have stem cell properties, and they're also highly abundant within the retina. What happens is that if you look at lower organisms such as zebrafish, when the retina is injured, the Mueller glial cells, the stem cells, can actually become other retinal neurons and regenerate the retina and repair the vision. And it's really a remarkable regenerative ability. Now, when you move up the evolutionary ladder to a chicken, there's also this regenerative ability for Mueller glial cells to repair vision. But as you move up to the mouse, this regenerative ability is really suppressed. When there's an injury to retina, the Mueller glial cells can be activated and divide and generate a small amount of retinal neurons, but that is not sufficient to repair and restore vision. And in human, this regenerative ability is being lost. So the general belief is that the Mueller glial cells have the potential to become other retinal neurons and regenerate the retina, but this ability has been suppressed or lost in higher organisms like human. So what we argue is that using a technology called cell reprogramming, we can stimulate the cells to regenerate new photoreceptors in patients to effectively restore vision. Cell reprogramming technology is really a Nobel Prize winning discovery. It's a technology that allows us to reprogram cell identity by controlling the genes in a cell. What you see on the right side is the illustration of the Nobel Prize discovery. We can now take a skin cell and switch on just 4 genes, OCT4, SOX2, KLF4, and c-MYC, and that would allow us to reprogram these skin cells into a pluripotent stem cell called iPS cells. Now, the key point here is that different target cells, if we want to reprogram into different cell types, would require a different set of reprogramming genes. And this technology, cell reprogramming technology, has been applied to regenerate cells in different organs in mice, including heart, the liver, the brain, and the spinal cord. And we are looking to apply this technology in the eye. So what we've done is, with the generous support from Retina Australia, we have developed a cell reprogramming technology that allows us to turn myeloid glial cells into photoreceptors. We have identified a set of genes that allow us to do that effectively. And our research is actually the first to show that we can make this work in human myeloid glial cells. Let me tell you how we come to this development of this technology. Now, this is the strategy that we took to develop this cell reprogramming technology. We argue that if we understand the genes that are expressed in the human myeloid glial cells and photoreceptors, we could then perform computational prediction to predict the list of genes that we need to switch on in order to reprogram myeloid glial cells into photoreceptors. We would then test this set of reprogramming genes first in the lab dish using human cells, and then move on to test them in animal models, in the rodents, in a disease model, to see if our treatment using the cell reprogramming technology can regenerate photoreceptors and restore vision. In doing so, we have created the most detailed human retina gene atlas. And this really gives us the knowledge of all the genes that are expressed in a single cell within the human retina. What you're seeing here is a representation of 20,000 human retina genes and the genes that are expressed, or 20,000 human retina cells and the genes that are expressed. The color that you're seeing represent a group of major retinal cell types. Now, using this data set, we have then subsequently performed computational prediction with 3 different algorithms to predict the reprogramming genes that would promote reprogramming into photoreceptors. This allows us to shortlist 9 different genes, and importantly, we chose genes that we know have a key roles in retina development. We then take these 9 genes and test them in the lab dish using human myeloid glial cells. But first, we have another problem to overcome, and that is we have to find a good way to switch on these reprogramming genes. So we've ended up spending a fair bit of time testing and optimizing a new technique called CRISPR activation system, and this allows us to switch on these reprogramming genes very efficiently, and we are able to activate multiple genes simultaneously. This is the paper that we published, and this is an example of how this technology works. When we switch them on, we're able to upregulate the genes to about 1,000-fold increase compared to before. Now, using this CRISPR activation technique, then we test them in the lab dish with human myeloid glial cells to see which combination of genes is the most efficient in reprogramming them into human photoreceptors. We've done over 150 experiments, and in the end, we've come up with an optimized way to generate human photoreceptors, and this is what they look like in red. We've done extensive characterization to show that the human photoreceptors are of good quality, and we're confident about their identity. We know they express the right protein by looking at different photoreceptor markers with immunocytochemistry and qPCR. We know the photoreceptors are functional and they can respond to light using electrophysiology analysis with multi-electrode array. And we also know that the human photoreceptor we generate in the lab dish are similar to the other ones we find in the eye. And that is done by using single-cell transcriptomic analysis. Having established this cell reprogramming technology in the lab dish, the next step that we are doing right now is to test our reprogramming genes in the rodent model, in particular in a rat model with photoreceptor degeneration. And this is the set of experiment we're working on right now. we're injecting the reprogramming genes into the eye of a rat model that normally have photoreceptor degeneration. And we wanted to see if our treatment here would help prevent the progressive loss of vision or even restore the vision in this rat model. We'll be looking for evidence for photoreceptor regeneration using marker analysis. And we'll be looking to do retina structure analysis using optical coherence tomography. And more importantly, studying the visual response after treatment using electroretinogram. So we're still in the process of working with this, and hopefully in a subsequent presentation, I'll be able to provide you with more updates. But this set of experiments is really critical for us to assess the efficacy and the safety of our reprogramming technology as a gene therapy to treat photoreceptor degeneration. And this is critical before we start moving on to testing our technology in clinical trial with patients. Now, the advantage of our cell reprogramming technology is that we are developing this as a gene therapy using AAV, and this is really a proven technology to deliver genes to the retina. We think that our reprogramming technology is more translatable to human compared to other reprogramming technologies because we have tested them in the human molecular cells in the dish. And finally, our technology is looking to regenerate and stimulate the stem cells within the retina to regenerate photoreceptors. And this could benefit multiple retinal diseases. Now, in particular, the immediate target target cohort we are going for are actually patients with late stage of retinitis pigmentosa, which is why the animal model we're working with is actually a retinitis pigmentosa model. But we do think that photoreceptor regeneration in general would be beneficial to patients with other retinal diseases as well, such as age-related macular degeneration and also Stargardt disease. And that's the end for my talk. So I do need to acknowledge the people that help with this project, including past and present lab members that work on this project. Many of them are very talented students and staff. And also we've got really, really good help from local and international collaborators bringing in different expertise to really make this project possible. Last but not least, we need to acknowledge the critical funding support that enable this research, in particular, Retina Australia for really helping us kickstart this project. We've also got funding support from Centre for Eye Research Australia, University of Melbourne, NHMRC, and very generous philanthropic support from the Cal and Rosie Day Foundation and Betty Gibson. And that's the end for my talk. Thank you.
Thank you so much, Ray. That's just wonderful. So exciting to see such a great project. I'm going to keep moving because our other speaker, Lauren, needs to get away soon. So we'll move on and then we can have some questions at the end. So thank you very much for that.
Great. Thank you so much for that, Sally. And it's an absolute pleasure to be joining you again today. I always really enjoy speaking with Retina Australia members and giving you a bit of an update. So apologies in advance. I just got a text message from my husband who's been in an appointment in Melbourne and has got caught in the exodus of people trying to escape the city. So he's not going to make it in time to pick up our son from kinder. So I've got the emergency message saying you need to do that. So apologies in advance. But look, I really just wanted to give you a bit of a short and sharp update of some of the projects that we're doing at the moment that have been very, very kindly funded and supported by Retina Australia. The first one I wanted to talk about was a study that probably many of you listening have actually participated in. So this was a survey that we've run this year looking at how people feel about retinal gene therapy. And so we sent this survey out through Retina Australia and Vision Australia and a few other avenues, including the Australian Inherited Retinal Disease Register. And what we're really trying to do is find out more about what people think about gene therapy. So Ray's done an excellent job of explaining what that is. But we really wanted to know what people's knowledge was at the moment and what they thought some barriers might be to them getting the treatment. And the reason that this is so timely at the moment is, as Ray mentioned, that Luxturna was just approved last year. So there's now a commercially available gene therapy for one of the inherited retinal diseases. And the next generation of gene therapies are really fast behind. And so we know these treatments are going to start becoming available. So we wanted to know more about how people who might benefit from them feel about these. And so we were completely ecstatic. So to give you an idea, we were hoping for 300 completed surveys. That's pretty standard around the world in these sorts of studies. We got over 730. So absolutely ecstatic with the results. What it's actually done is also sparked a lot of interest from some of the pharmaceutical companies who are also very interested in finding out about what people think about these treatments. And so we're hoping that that will actually spark some future funding opportunities for the research and some more collaborations. So we are now going to spend quite a number of months going through that data. So if any of you did do the survey, you'll be aware it was over an hour to do. So there's a lot of data in there. So we're going to do some analysis, and then we'll hopefully report back to you later this year. So this project was headed by Associate Professor Heather Mack and myself, and it also included people from Perth, so Fred Chen, many of you will know, and John Grigg from Sydney. So it's very much a nationwide survey, and we're very excited to share those results with you later in the year. The other thing that this survey did was actually bring to our attention that, or something that we knew, but really reaffirmed that many people have not yet had a genetic diagnosis. And this is for a number of barriers. So obviously it's expensive to get genetic testing done. Often ophthalmologists and eye care practitioners aren't offering these tests or aren't really expressing that they're now quite important to do. So our next step is actually to do an audit of clinical files, and we're going to start with some private ophthalmology practices and then hopefully move to the public system, so the Eye and Ear Hospital down the track. And we really want to go through and work out how many genetic diagnoses are there and get a little bit of an idea of the landscape of retinal disease in Australia. So this is, again, quite exciting, and this is sparked from that initial Retina Australia funded project. The other project that's being currently funded by Retina Australia is one that I'm leading on sensory substitution devices. So apart from being a complete tongue twister, what these are low vision aids which use our other senses apart from vision when the vision's not doing so well. And so these are things like tactile devices that might give you a buzz on either your hand or your back or potentially on your tongue to show you that you're coming up towards an obstacle, or audio cues as well. And what we're very interested in is taking these from where they are at the moment, which is very much really research-based devices, and turning them into something that's more useful for people. So we are working with a computer scientist called Dr. Chris McCarthy, who's at Swinburne University, and his expertise is in vision processing. So he's able to take a very complex scene like we see in our normal environment and pick out the parts which are very important for us for things like mobility and activities of daily living. And one area where he has an international reputation is in depth perception. So we are going to use his programming skills to optimize some of these sensory substitution devices so that they work better and so that they're a bit more useful for things like mobility. So this project is being run with a PhD student whose name is Jerry. And for any of you that are on social media, you might have seen a video we did a couple of weeks ago talking about this project. And so Jerry's going to work with my team and with Chris's team and Matt Keto from the Bionics Institute to try and bring together these different aspects and make these sensory substitution devices a little bit more effective. So what we've been doing so far is we actually are designing a new mobility course. And so this is in conjunction with the Department of Physiotherapy at Melbourne University. And so we're setting up a course where we can actually assess these devices to see, you know, how they work and how we can make them better. But it's also a project in itself, to be honest, because up until now, a lot of these mobility assessments are done in, you know, hospital corridors or small rooms that we can find. And again, some of you on the call today have probably done this sort of research with us before. And so moving it into a physiotherapy lab actually gives us more measurement opportunities. So we can do things like looking at force plates on the floor that measure how fluently somebody walks through an obstacle course. We've got cameras all around the room so that we can film from every different angle. And so there's a lot more, I guess, scientific numbers and things that we can come up with by moving into this sort of lab. The other aspect that we've been working a lot on in the last few months is looking at using virtual reality for some of these measurements as well. And this is quite exciting because this means that we might be able to do similar sorts of assessments in, for example, someone's home, rather than having to bring them into the university to the lab. So the project's going really well. We're aiming to start data collection on the actual mobility course and the devices in the next month. The other thing that's come from this is Jerry has just launched a survey looking at what people have experienced in terms of mobility aids. And again, Retina Australia have been incredibly supportive to us by putting that up on social media. And so if you haven't seen it, please let me know. I'm happy to forward that link to you as well. And the idea with that is we'd like to get an idea of what mobility aids people use, what they like about them, what they don't like about them, and how we might be able to improve with this next generation of device. Now, the last thing I thought I'd mention, because it's often a question we get at the moment, is how are things going with the clinical trials? So again, Ray's done an excellent job of letting you know how gene therapy works and the fact that there is one commercially available treatment. But obviously, we're at clinical trial stage for a whole lot of other treatments. And so some of you may have seen on the news about 2 weeks ago, we made an announcement that we've started doing gene therapy at the Centre for Eye Research Australia for people that have age-related macular degeneration. And these are people that have the dry form of AMD, and it's a gene therapy which is trying to slow down the progression of the atrophy. So it's very similar in terms of the method that's used, in terms of how we assess people pre and post treatment, as it will be for conditions like retinitis pigmentosa. So this is very exciting for all of us, really. So we obviously have the processes set up in place now to run these clinical trials. So we have the pharmacy on site, so we're able to actually mix up the treatments and then provide them through the surgery. And our surgeon is someone that most of you probably know quite well too, that's Dr. Tom Edwards. Again, works with Ray too. You've noticed we're quite a small little world. And so yeah, Tom has done these first surgeries for this gyroscope gene therapy study. And we're now talking to a number of pharmaceutical companies about what the next trials that we will host will be. You're probably aware there's basically a lot of work going on now into different gene mutations and which ones will be the next to have treatments developed. At the moment, we think that the most likely candidates for the next wave of trials will be obviously RP65, which is the one that is for Luxturna, but choroideremia, the X-linked RPs, retinitis pigmentosa, is likely to have trials coming up. And then in particular, that's for a gene called RPGR. And the other that the companies are really knocking on our door about is achromatopsia. But I guess that really ties back into the other large project that we're doing, which is this natural history study. And so we are obviously trying to get in everyone that is interested in research to find out what genetic mutation they might have, what their eyes look like, what their vision's like, so that number one, we can learn more about the conditions, but number two, we can then also have a database of people that are ready for these trials. So to give you an idea of how it works, when a pharmaceutical company reaches out to us to say they'd like to run a trial in Melbourne, We then talk to all of our collaborators, so including the Australian Inherited Retinal Disease Register in Perth, obviously all of the doctors that work in IRD and our different research sites across Australia, and we get an idea of how many people have that particular gene. And so obviously, the more comprehensive our databases are, the better, because we can then say to the pharmacy companies, you know, we have X number of people ready to go, and they're more likely to bring the treatment to Australia. So we're going as quick as we can. We had really sped up in the last couple of months, but obviously this new lockdown is going to slow us down again. You may, if you're involved with our research, you might start hearing from a couple of our newer team members. So Cece is a postdoctoral researcher who's working full-time on the project, and Asena is a PhD student that's starting with us as well soon. But our core group, who many of you know, so Tom Edwards, Fleur O'Hare and myself, still very much involved in this natural history study. I've got a couple of contact details just up now on a slide, which obviously I'm happy to supply to everyone in any way you'd like. We do have an email address, which is ird@groups.unimel.edu.au. And there's a phone number here as well. But look, obviously, if you're not able to make that out on the slide at the moment, either Sally or myself can provide that to you too. But we are always incredibly grateful for any help that you know, either Retina Australia or the members can provide to the research. And hopefully that gives a little bit of an update on some of the work that you know, obviously your fantastic fundings let us do over the last year. So thank you very much.
Okay, so now what we need to do is go to our attendees and see who would like to ask us some questions. Okay, well, I'll ask one. I wanted to ask you, Ray, you mentioned that one of the things that you were, when you start to do testing with people, you're looking at late stage RP patients. Can you talk about why you would use that as opposed to people who are perhaps not as far advanced?
Yeah, sure. So I guess you know, with in principle, actually the technology we're developing would be suitable for late stage and also early stage or intermediate stage of photoreceptor degeneration. But we did have a discussion with Tom Edwards, and the feeling is that in terms of developing a clinical trial, it would be a lot simpler and more feasible to start with late stage retinitis pigmentosa patient first, when you know there's really more extensive loss of vision. You know, that's possibly where we can see a better improvement for vision in the first place. But in principle, it actually should be beneficial to even early stage or intermediate stage. Yeah.
So that's something that in the future potentially you could open it up to people who are in an earlier stage.
Yeah, absolutely. Yes.
Great.
I might just add to that as well, actually. I think this is one of the really exciting things at the moment is that there are all of these treatments for the whole spectrum. So, you know, obviously Ray was mentioning that his approach can be sort of, you know, early through to late. There's obviously still work going on vision prostheses or bionic eyes, and that's very much for people that have late vision loss. Gene therapy tends to be more early to moderate disease. So we think that you need to have at least sort of 60% of the photoreceptors still there for gene therapy to work. And so this is why there are so many approaches. People often say, you know, well, why haven't you picked one, I think, for the field? But it is because, you know, we need all of them.
Fantastic. Thank you so much. So now we've got a question from Leighton Boyd.
Ray and Lauren, one of the questions that has certainly been out in the media, and we talked about it before, but is some work out of Sydney. I think Save Sight, I believe, is doing some work in the retina space. Do either of your groups, and I think mental block, John Grigg is involved is he involved in that work up there, and how closely are you guys and your teams working with them in that space?
Yeah, I might jump in here. Yeah, so very closely is the answer. So the survey that I mentioned before that Heather's has been running, that was funded by Retina Australia, John's one of the lead investigators on that too. So it's John, Fred, Heather, and myself. And we, as I mentioned before, you know, if we are interested in a particular form of inherited retinal disease, all of the groups around Australia will work together to try and you know, maximise the reach that we can have to people. There is a group called TOGA, and I can never remember what it stands for, but it's basically researchers and clinicians that are interested in retinal disease across Australia, and we all tend to meet every sort of 2 to 3 months. And that's a really powerful thing because it means that everyone is aware of what's happening in other states, and we're able to sort of share our expertise and our results.
Yeah, and for us, we work with 2 groups in SafeSide Institute. So Mark Gillies is a very good collaborator, and he really is, you know, brings in tremendous expertise in the molecular cells in particular. And we also work with Ulrich, actually, with the human Retinogen atlas. She was actually part of the team as well. So yeah, obviously, you know, I think, you know, the retina researchers within Australia working, collaborating interstate, and, you know, trying to address these issues. Yeah,
That's great.
Yeah.
Thank you so much. I've got a question here from Jane Cherry. She has a question for Ray. In terms of testing for the gene therapy, is there an expected amount of time the testing would take place with animals before moving to human clinical trials, or is it dependent on the results from the testing?
Yeah, so obviously I think it would be dependent on how the result turns out. If it's smooth, we actually have a timeline of a 5-year plan to take our technology to a clinical trial. We're actually seeking a commercial approach and investment to try to speed it up to about 3 years. And, you know, the idea being that with more resources and manpower, we could actually accelerate the research. But at the moment, this is all, I guess, at the early stage. And we really need to work hard to make sure that the technology is effective and, more importantly, safe before we actually move to the clinical trial. Yeah.
Absolutely. That's fantastic. I just wanted to make one comment just in relation to the survey with your mobility course that's happening, Lauren. You mentioned that we'd sent some information out to members on social media. There will also be a mention of that in our upcoming June newsletter, and I'll put a link there for people so that for the people who are not on social media, you can find that that way. So.
Fantastic. And I should just say that survey is very, very short. It's very different to the one we did on gene therapy. So this one's only about 5 minutes.
Fantastic. All right. Does anybody have any further questions? This is your opportunity now. Leighton Boyd again.
Well, you know, when you're the chairman, you can raise your hand as many times as you like. No, I was more interested in the funding for all these things. I mean, Retina Australia, we try to do what we can do, the limited resources, but the involvement with pharmaceutical companies across the world seems the way, and they've certainly got lots of money and are utilising it. So do you guys find them? I've mentioned, I mean, a few, you mentioned in your presentations about working with various pharmaceutical companies. Does that work well from where you people come from?
So you're right. It's definitely starting to happen more and more now. So probably what is so valuable about the Retina Australia funding is it gives us proof of concept data that we can then go to the company. So obviously pharmaceutical companies do not care at all about our theories. They just want to see proof that what we're doing works. And so that's where it's really invaluable to be able to go with them after having some initial funding. One thing that we are pushing really hard now with the pharmaceutical companies is to try and help support genetic testing for people with inherited retinal disease in Australia. So that's obviously really important to them so that they know how many people might want their treatment. And so we are getting quite a lot of traction with that at the moment. So the companies are starting to see that that's something really important. So that's probably a really big thing from my point of view, is trying to get them to support some of that testing. Yeah, and then I'll let Ray sort of talk more about the treatment side of things.
Yeah, sure. I mean, I will second Lauren's comment. I think, Retina Australia's funding is really critical as a seed funding for us to kickstart the project. Many other funding organizations, including commercial companies or NHMRC federal funding, really they're not really strong supporter of seed project. And this is where Retina Australia is really helping us to get the set of pilot data so that we can go and apply for larger funding to really follow and develop our technology through to clinical trials. So absolutely critical to our research, and we really appreciate that. Yep.
Can I just have a follow-up, Sally, please? Retina Australia, we believe, and we're working very closely with Retina International about the genetic testing, because it is so important that probably the most commonly asked question is, yes, I've got this disease, but IRD, but how's it, what genes it from, and all of those issues. So we've been working very closely, the board and myself, and with some pharmaceutical companies. Retina International, one of their priorities is to get, hopefully, free genetic testing, even get a resource that people throughout this country and even the world will know exactly where to go and how much, where to go, what place, wherever you are. And so that it is important. And in fact, only last night we're on a meeting with a pharmaceutical company discussing various aspects of this thing. So I hope that, sooner rather than later, there'll be these resources that all of us as patients and will be able to get a genetic test, get a result, and then you people as the scientists will do all the hard work to find a treatment and cure.
No, I think you're exactly right. And and I think one thing that I think I often tend to forget a bit is, this has all happened so quickly. So the first genes for IRD was sort of only really, 1980s or so. And so since then, it's gone from a handful of known genes to over 300 known genes. And so the opportunity now is so exciting, because we can actually pinpoint. I think the most recent data suggests about 85% of people with an inherited retinal disease, we can find the gene. And so it's just so important. And so the work that Leighton just mentioned with Retina Australia, working with Retina International, is so important.
That's fantastic. Thank you very, very much, both Ray and Lauren, for your time today and sharing that exciting information, because it is so great for us to hear about these positive advances. So I think at this stage, I'd like to say thank you very much to our presenters for your time, and thank you to all the attendees. And I hope that you found this a very useful webinar. So thank you very much, and have a good afternoon.